Listen-only mode.
Hello and welcome to the first webinar in the 2017 Pulse webinar series. My name is Glenda Cleasy and I am the Agronomy Specialist with SPG. With me is Andrea Lauder, Communications Manager with SPG. Andrea is the webinar organizer and is looking after the functioning and administration. Thank you, Andrea. To let you know, we have 3 additional webinars scheduled, and the additional webinars can be found on the SPG website under News and Events tab. Topics that we'll be covering in additional webinars include soybean agronomy, scouting for diseases and pulses, and basics of grading your pulses. CCA and CCSC credits are available. To get credits for today's webinar, you must be watching it live for CCA credits. For those who attend the webinar, Andrea will send out an email after the webinar requesting your CCA number. At this time, please respond with your number. If more than one person is watching from one computer where you're at, you will need someone to verify those in attendance and can include all CCA numbers of the participants who watched live. Webinars will be recorded and posted to the SPG website for future viewing for those unable to attend or for those of you who want to look back at the material that was covered. Recordings will be posted to the SPG website under the Communications tab. For today's webinar, all participants will be muted, but we are very happy to take questions. To ask a question, please type it into the question box located in the GoToWebinar dashboard. You can send questions at any time, and we will be holding the questions until the end of the presentation. Today's speaker is Jeff Shano. Jeff is a professor of soil fertility and professional agrologist who works in the Department of Soil Science at the University of Saskatchewan. He holds the Saskatchewan Ministry of Agriculture Soil Nutrient Management Chair in the College of Agriculture and Bioresources and is a Fellow of the Agricultural Institute of Canada. He was born in Saskatchewan and completed his undergraduate and graduate degrees in the College of Agriculture at the University of Saskatchewan. Jeff is going to speak to us today on pulse macronutrient requirements and contributions. Thank you so much, Jeff, and I'll now turn the presentation over to you.
Great. Thanks a lot, and good afternoon to everyone. As we move into this busy spring seeding time coming up, I'm going to talk to you folks a little bit about pulse macronutrient requirements and contributions. And when we think about pulse crops, grain legumes in other words, and their contribution to soil fertility, folks, of course, really where it all starts is right there, and depicted there on that pulse crop, we see nodules there on the root, and of course those nodules are the home for the rhizobium bacteria that when we inoculate with a superior strain of rhizobium that comes from a commercial inoculant, of course that rhizobia then can fix nitrogen for the legume crop itself and also contribute to the nitrogen nutrition of the crop that follows. So very important component of the whole fertility aspect of pulse crops. And this slide here, which I borrowed from Dr. Fran Wally, who has worked for many years in biological nitrogen fixation. I really like this slide because it shows the nature of the symbiosis, the two-way relationship that exists between the legume plant and the rhizobia that lives in those nodules on the roots. And it's a two-way relationship or symbiosis because in order to carry out nitrogen fixation and live in those nodules, the rhizobia needs an energy source, and that comes from the plant. So the plant sends energy to the nodule, and then the rhizobia in that nodule convert atmospheric nitrogen, N2, and the air is 78% nitrogen, so there's lots of N2 in the air. The rhizobia convert using enzyme that atmospheric nitrogen to plant usable nitrogen, and then that nitrogen is transferred to the plant. So it's a symbiosis, a two-way relationship, and we often refer to biological nitrogen fixation as obtaining nitrogen for free from the air. But recognize, folks, in fact, that nitrogen fixation is energy expensive. That is to say, there is a cost to the plant. The plant has to give up some energy for that process to take place. And indeed, when we think about things that affect nitrogen fixation, really anything that affects the plant is going to affect fixation. So environmental conditions, available nutrients in the soil, and so on, which we'll talk about in just a few moments. And indeed, the yield of the crop can give a pretty good idea of nitrogen fixation, the amount that is derived from biological nitrogen fixation, as we'll see in a couple of moments as well. So a question, how much nitrogen is derived from the air and converted into plant nitrogen by a legume crop? And indeed, it's variable, and there are different ways to measure that nitrogen fixation. A common method that is used is nitrogen 15 isotope dilution technique. Different ways give different numbers. But any way you slice it, folks, that biological nitrogen fixation is a significant contribution. And generally, anywhere from 50% to 80%, sometimes even higher, I've seen 85, even 90% of the nitrogen, in some cases, in that legume plant, in that pulse crop, comes from the atmosphere by a biological nitrogen fixation in that nodule. When we look at a nodule on a legume root, as shown here, and we slice open that nodule, in this case we see that nice, bright, beefy red color that indicates that those nodules, the rhizobium inside, are actively fixing nitrogen. The red color comes from the presence of leg hemoglobin, which is kind of similar to the hemoglobin in your blood that binds oxygen, gives that red color. What that leg hemoglobin is doing is it's helping shield that enzyme from oxygen and allowing the enzyme nitrogenase to carry out the nitrogen fixation process and convert that nitrogen into plant usable N. So we can see there bright red nodules there indicative of active nitrogen fixation. So how much nitrogen is derived from fixation in Western Canada? And we can look at this in terms of ranges for different legume crops. The forage legumes like alfalfa and clovers tend to come out on top. So it's not uncommon to see a good alfalfa crop fix around 200 pounds of nitrogen per acre in a year. Some of the pulse crops are not too far behind that. Faba beans, for example, a range of 80 to 160. In fact, some data that we obtained from the field last year might suggest that we might even see higher amounts than 160, perhaps even as high as 200 pounds of nitrogen per acre derived from fixation in a faba bean plant. So they're very good nitrogen fixers. Not too far behind are peas, and then soybeans with proper inoculation, as I'll talk about in a few moments, can also be significant amounts of nitrogen fixed by that crop. Lentils, variable as well. High-yielding lentil crops also can fix a lot of nitrogen, as can chickpea. Dry beans tend to be on the lower end of the nitrogen fixation scale. Overall, when you look at those amounts of nitrogen derived from biological nitrogen fixation, of course, that represents a significant dollar value when you look at it in terms of the cost to purchase that nitrogen if you were buying it as commercial nitrogen fertilizer. And of course, the actual amount that you see depends on a lot of factors like inoculation, nodulation, and indeed that's where your commercial rhizobial strains come in that you purchase that are superior nitrogen fixing strains of the rhizobium. Environmental conditions very important. If it negatively affects plant growth, it's going to negatively affect the amount of nitrogen derived from fixation. Soil available N, that I'll talk about next, and other nutrients like phosphorus. Phosphorus, of course, is the energy currency of life. So we said that nitrogen fixation is a very energy-intensive process. So without phosphorus, we're not going to get nitrogen fixation taking place. I want to say just a few words about soil available nitrogen and fixation. If the available nitrogen level in the soil is high, that is to say, for example, lots of nitrates in that soil because it was fallowed or maybe received an application of nitrogen fertilizer, a high rate, or there's residual nitrogen fertilizer left behind from the year before, folks, the legume will use that soil available nitrogen instead of allowing fixation to occur. And that's because the fixation, as I mentioned before, requires some energy. lazy on behalf of the plant. So the legume will be lazy, and if there's available N in the soil, it'll use it rather than fixing it. So as shown there, 2 pea plants that I had in different growth media, one with no nitrogen and one with nitrogen fertilizer added. Both those pea plants look pretty good. They're both doing well. You'll notice that the plant without the nitrogen fertilizer, you can see nodules on the roots. So in that case, the bulk of the nitrogen in that pea plant came from fixation, where in this case, where it was available in the soil, no nodules there, the plant didn't allow the fixation to occur. Instead, it used the available N in the soil. So if there's lots of nitrogen in the soil, the legume crop will generally do fine. But however, what you're missing out on is that external input of nitrogen from the air for free, so to speak, from that biological nitrogen fixation process. Certainly, sometimes when related to nitrogen fertility, fertilization, and pulse crops, a bit of a debate. Starter nitrogen or no starter nitrogen fertilizer for pulse crops. And indeed, if you go into the literature, you can find examples where there was some benefit from starter nitrogen in terms of yield, for example. In other cases, no benefit. Depends on the crop. I think depends on the environmental conditions. Overall, I think I would say small amounts of starter nitrogen, talking here less than 20 pounds of nitrogen per acre for pulse crops, may be beneficial, can potentially be beneficial. Examples of that, those types of situations would be on highly nitrogen deficient soil, an example where you've got less than 10 pounds of nitrate nitrogen in the top 12 inches. Another example might be where you've got a lot of surface low nitrogen residue like cereal straw, high carbon to nitrogen. If that's incorporated and immobilizes a lot of available nitrogen that coincides with just about the time when that pulse crop is germinating and emerging, that may be a factor where a little bit of starter N may be beneficial to help carry the plant through to the point where the nodules are established and nitrogen fixation can begin. And indeed, I think any time where the onset of nitrogen fixation may be delayed. For example, by cool temperatures, wet soil conditions, also disease pressure that might be an interfering factor with the onset of nodulation and nitrogen fixation on the roots of that pulse crop. I want to talk a little bit next about a study recently completed by Jing Zi, PhD student in the soil science department at the U of S, and Jing successfully defended her thesis this past January. And in this study, we looked at yield, nutrient uptake, nitrogen fixation, and release by 3 different pulse crops we had in our comparison: soybean, pea, and lentil, and also the impact on yield and nutrition of following crops in rotation. So a little bit about this study. We have 4 field sites in this study, 2 in the dark brown at Saskatoon and Scott, and 2 in the black soil zone at Rosthern and Yorkton, as indicated here. So in this region of Saskatchewan here is where we're working. And in this experiment, in this study, we had 3 different varieties of each of the pulse crops, the legumes, 3 of pea, as indicated there, representing different market classes, 3 of lentils, so we had large green, small red lentils there, and also 3 different short season soybean varieties, and we also had wheat in there. Wheat we had in there because we wanted to have a non-nitrogen fixing crop that we could use to evaluate how much nitrogen was derived from fixation by the different pulse crops in our study. So this study began in 2014, and to give you folks just a bit of an idea of what the available nutrients were at our 4 sites, a couple of sites had higher nitrate levels, a couple of sites had lower nitrate levels in the top 6 inches. All generally had good phosphorus fertility, pretty good phosphorus fertility as indicated there. Sulfate was also pretty good. There were subsoil sulfates. The Saskatoon site was an interesting one because it was one that was actually located in a depressional area with a fairly high water table, and so we did have some sulfate salts present there as indicated by the elevated electrical conductivity and the salts in Saskatchewan that cause salinity or sulfate salts. So associated with that slightly higher electrical conductivity, lots of sulfate present there right at the surface of the soil at our Saskatoon site. So this is the yield that we got in 2014 at our 4 sites, Rosthern, Saskatoon, Scott, and Yorkton. And we've got lentils, the 3 varieties of pea, and the 3 varieties of soybean. In terms of differences among varieties, generally what we saw was mainly manifested in the lentils, where the red lentils tended to yield the highest, as shown there. In terms of the crops, pea yields tended to be higher, the highest, especially at this Rosthern site. At Saskatoon site, we see their yields were generally lower because of the salinity pressures that were encountered at that particular site because of the poor drainage. You'll note here at our Scott and Yorkton sites, our soybean yields of our 3 varieties were pretty darn close to what we saw from our peas at those 2 locations. So generally, peas tended to be on top, greater than or equal to soybean, greater than lentil. Now, when we looked at the nitrogen contained in the above ground plant components, that is the nitrogen, what we call the nitrogen yield, the nitrogen in the grain and straw, that pretty well followed what we saw in terms of yields, highest in the peas here at the Rosthern site. But overall, as we can see here at a couple of sites following the yield patterns, our soybeans also tended to be really quite close in terms of the amount of nitrogen in the above ground material to our peas, which were in turn higher than the lentils. And indeed, the amount of nitrogen in that above ground material, particularly in these peas at the Rosthern site, which yielded very well, 60 plus bushels per acre, we had over 200 pounds of nitrogen per acre in the above ground material in the peas at that site, around 150 for the soybeans here. Now, in terms of the proportion of that nitrogen that came from biological nitrogen fixation, what we call percent nitrogen derived from the air, we can see here typically 50 to upwards of 70% of the nitrogen in our pulse crops came from the air through biological nitrogen fixation, and the soybeans actually had some of the highest percent NDFA values, higher than, for example, in this example, higher than pea and pretty close to what we saw for lentil. So the soybeans are good nitrogen fixers and did derive a significant proportion from the fixation. Now, it's important to note that in the case of the soybeans that we grew, we did double inoculate. We applied double the rate of Rhizobium japonicum inoculum. And the reason why is because, in fact, the Rhizobium japonicum populations, the indigenous populations in prairie soils are very low. So it's important that soybean be inoculated with that correct species, Rhizobium japonicum, in order to get the maximum amount of biological nitrogen fixation and to achieve these high percentage of nitrogen in the plant that's derived from the air. So in particular, comparing those, and not a lot of data on soybeans in relation to other pulse crops, we found that the modern short season varieties have good potential for yield and also nitrogen fixation. Now, some more recent work conducted in a project that began this past year by a graduate student, Serena Klippenstein, in the Soils Department here at the University of Saskatchewan, looked at faba bean yields at 4 sites and showed that faba bean, from the 2016 data, also a very good yielder and nitrogen fixer. So in this case, we had faba beans at 4 different sites: Saskatoon, Outlook, Rosthern, Meath Park. As you can see by the average grain yield, 6,000 kilograms per hectare, we were achieving 70 to 90 bushels per acre of those faba beans at 3 of our 4 sites, very good yields. Our Outlook site, it was suffered from excess moisture, so our yields were lower. In terms of response to fertilization, non-fertilized in the orange, fertilized with phosphorus, potassium, sulfur, indicated there by the blue bars, we didn't see a big response to fertilization of the faba bean. Now, the sites that we had were similar to the sites that were used by Jingzi in that they had pretty good phosphorus fertility, not too bad sulfur and potassium, and good potassium fertility as well. So we didn't see a big response at all there. In fact, non-significant response to fertilization in 2016. And we're going to be repeating this again in 2017 at another 4 sites. Looking at the grain and straw yield of those faba beans, and this is the average of 4 faba bean varieties, the nitrogen yield depicted for our 4 sites. The nitrogen contained in the grain of those faba beans, as you can see, is significant, much greater compared to the nitrogen remaining behind in the straw. And when we look at the nitrogen yield, the amounts of nitrogen there in the above ground material, we're approaching 300 kilograms of nitrogen per hectare, and a very large proportion of that contained in the grain. So, you know, if we make an estimate of, say, 70% of nitrogen derived from fixation by that faba bean, and we're going to be using some data from Dr. Diane Knight, as well as some assessments of nitrogen fixation on faba beans under controlled environment to see just what proportion of that nitrogen is, in fact, derived from the air, you can see where we can look at some very significant amounts of nitrogen coming into that system from biological nitrogen fixation. Since it's very much biased towards nitrogen in the grain component there, you can also see that a lot of that nitrogen gets removed when we harvest that faba bean from the field as harvested grain, faba bean grain. Which I guess, folks, brings us to the next topic is, you know, what's the nitrogen contribution of a legume to a following crop? Recognizing that it can be significant, but it's not typically huge if the grain is harvested and removed. And that's because, as shown in that previous data there for the other pulse crops, and indeed the seed contains typically about 70% of the nitrogen in a mature pulse crop, and it may indeed even be higher than that, as shown with some of our data from the faba beans. So if we assume that the seed contains about 3/4 of the nitrogen in a mature pulse crop, recognize that this amount of nitrogen that's removed in the harvested seed is about the same as what comes in from the biological nitrogen fixation. We showed data there, maybe percent nitrogen derived from the air, around 70%. So what we have then is a kind of balance where, in general, where the nitrogen that's removed in the harvested pulse seed is about equal to what's coming in from fixation. And of course, that fixation, we may consider it to be coming for free from the air through the activity of the rhizobium in the nodule. Whereas, of course, in a non-legume crop, the nitrogen that's contained in that plant and that's removed from the system in grain harvest has to come from the soil and the fertilizer. So when we think about benefits to the following crop of having a legume, we maybe first think about nitrogen benefits, but there are both nitrogen benefits and non-nitrogen benefits. And a value that's sometimes calculated is what's called the nitrogen fertilizer replacement value. And what this is it's an experimentally derived value, a calculation of how much extra fertilizer nitrogen would it take to bring the yield of a non-legume crop, like wheat, growing on a non-legume stubble, like let's say its own stubble, wheat stubble, to the same, or flax stubble, to the same yield as when that crop is growing on legume stubble. And so, as an example here, I used some data that was collected a few years back at Melfort, Saskatchewan, published by Wright in 1990, looking at barley response to added nitrogen fertilizer. So yield response to different rates of nitrogen fertilizer growing on different legume stubble, faba bean, field pea, and lentil. We see the response there. Not a lot of difference among the different stubble types, compared to barley growing on barley stubble. And there we see lower yield of barley growing on its own stubble. As we add nitrogen, the curves kind of start to converge together, but they still remain apart. And they remain apart because what you have there is both a non-nitrogen benefit, that is, benefits of growing barley on the legume that really don't have anything directly to do with nitrogen, maybe related to improved root health, reduced susceptibility to disease, and a direct end benefit. Ultimately, what is predicted to happen, if you add enough nitrogen, those curves will converge together. And this is called the nitrogen fertilizer replacement value. How much did you have to add to get those curves to come together? And this can be quite a significant amount of nitrogen. So in work that was done by Paul Jefferson, and I was involved in this study as well as others, looking at nitrogen fertilizer replacement value of legumes, an example here in this case, 2 years of alfalfa or 1 year of pea versus flax stubble to a following wheat crop. What we see here is some pretty significant nitrogen fertilizer replacement values or equivalent. Alfalfa study, 300 kilograms of nitrogen per hectare, pea stubble, 180 kilograms of nitrogen per hectare, and generally as the environment got drier in this study, as we moved from brown to dark brown, for example, that nitrogen fertilizer replacement value did tend to diminish. So the greatest effect of nitrogen fertilizer replacement value of growing that crop on that legume stubble came in the more moist environments. Under really dry environments, having a forage legume in there indeed could be detrimental to yield in the immediately following year because it tended to dry out the soil profile. In our study where we were looking at the availability of nutrients and growth of crops on our three different pulse crops, lentil, pea, soy, and we also had wheat stubble in there as well. You can see here in this picture, wheat growing on wheat stubble here just doesn't look as good as, of course, the wheat growing on the different pulse crop stubbles, soybean, pea, and lentil. Part of that is related to lower nitrogen availability there on the wheat stubble, but also some other non-nitrogen effects as well, like disease, for example, pressure, root health, and other factors as well that contribute to that, as shown there, less thrifty wheat when it's growing on its own stubble. So one of the things that we were interested in this work was what was the effect on nutrient availability of those three different pulse crops, soybean, lentil, and pea, to a following crop. And so in this study we had wheat and we had canola at our sites, and we looked at availability of nutrient on the different crop stubbles using PRS, plant root simulator probes placed in the field, in the plots, and looking at supply over the season, as well as looking at the yield and nutrient uptake by the crops. Wheat in this case is shown here, also canola at our sites, looking at the uptake of nutrients by the crop. And this is an example of what we saw at two of the sites, Rostern and Saskatoon, where we looked at the cumulative supply of available nitrogen released over the season on lentil, pea, and soybean stubble. The lentil is the blue, the pea is the red, and the soybean is the green. In fact, at our sites, we didn't see much difference in release of available nitrogen over the season as measured by PRS supply rates in our different pulse stubbles. We were a little bit higher from the lentils here early on, reflecting a lower carbon to nitrogen ratio at the Rostern site, but then it tended to go down a little bit later. Overall, in the end, not much difference, no statistically significant difference, and even less difference here at our Saskatoon site. And indeed, when we looked at the yield of the following crop, this was the grain and straw yield of wheat. We had canola at our Yorkton site that was growing in 2015 on the different crop stubbles. In fact, the wheat yield, and here we got our grain yield and our straw yield on top of that, so that's the total yield on soybean stubble, pea lentil, or pea stubble, lentil stubble, and then wheat on wheat. We see not much difference in the yield or the nitrogen uptake of the wheat that was growing on the different pulse stubbles, but was significantly lower when the wheat was growing on its own stubble. We didn't have any wheat growing at our Scott or our Yorkton sites, and that's why you don't see any bars there. But no difference among the pulse stubble types. So folks, what we found in this work at our four sites is really that our soybean, our pea, and lentil generally had a similar effect on nitrogen availability and yield of the following rotational crop, which certainly does indicate, at least based on the results that we obtained, that there would be no real great need to adjust fertilizer nitrogen rates according to which type of pulse crop you would have had growing in that field the year before, if you were going to be growing canola or wheat that year. So that's nitrogen, folks. What about phosphorus? Certainly the thing about legumes is that they do have a rather effective root system for getting nutrients out of the soil. Indeed, those legume roots support a strong colonization of beneficial AM fungi, arbuscular mycorrhizal fungi, that increase the root surface area, the water, the nutrient uptake ability of that crop. Also, legume roots can acidify the root zone and thereby solubilize these calcium phosphates that are common in prairie soils, and that's certainly something I think we've observed in forage legumes that can root down deep and bring up some of, solubilize and mobilize some of that insoluble calcium phosphate present as apatite in the subsoils at depth. And I guess overall this probably explains why pulses are sometimes not highly responsive to phosphorus fertilization unless the soil is quite phosphorus deficient. And that's because they're pretty good scavengers of phosphorus from the soil. And indeed, we recognize that pulse crops do have different tolerances to seed-placed fertilizer phosphorus. So with about a 10 to 15% seedbed utilization, for example, a 1-inch spread on a 9 or 10-inch spacing, are maximum safe rates according to the provincial guidelines for pea. Peas are quite sensitive to seed row placed fertilizer, so 15 pounds of P2O5 per acre as map in the seed row. Lentil and chickpea, a little bit more tolerant, 20. Actually, faba beans we find are quite tolerant to phosphorus placed in the seed row as monoammonium phosphate. And finally, soybean, not a lot of research work in Saskatchewan. We did a little bit in controlled environment work, and what we found was about 20 pounds of P2O5 per acre in the seed row with the soybean as monoammonium phosphate was about the limit before in our study we started to see some reductions in emergence of the soybean. Now recognize as well when we're thinking about crop removal of phosphorus, for some of these crops like peas, the crop removal as P2O5 is about 30 for pea and soy about 20 for lentils. So for some of the pulse crops, what is going to be removed by the crop is higher than what we can safely put in the seed row. So if you're looking at keeping things in balance, putting into the soil what you're taking out over time, if you're looking at maintaining that phosphorus fertility, for some of these crops that have high sensitivity to fertilizer phosphorus in the seed row, you're also looking at putting some phosphorus additionally in a band, for example, or perhaps adding it to the crop that you're growing before, which has a higher tolerance for phosphorus placed in the seed row, if you're interested in putting back in what you're taking out of the soil over time. And indeed, pulses, grain legumes, can be responsive to pea fertilization. Just showing here some data for soybean at a site at Central Butte. This was a soil that tested low in available phosphorus. It was up on an upslope area of a field that had not been particularly well managed in the past, hadn't had much of a history of pea fertilization, so a low available P level. And what we did see with the soybean, a significant yield response to in-soil placement of phosphorus, and that included placement in the seed row, placement below the seed in a band, was superior to where the phosphorus was simply broadcast on the surface and left on the surface. That placement approach really didn't result in any yield benefit at all over the control. In terms of phosphorus requirements, in our study where we were comparing soybeans, peas, and lentils, looking at the phosphorus contained in the above ground, the grain and the straw, and this is kilograms of P per hectare. So multiply this by 2.3 if you want to get kilograms of P2O5 per hectare. So here we're looking at those high yielding peas at that Rosthern site, contained in the above ground material, close to 50 or 60 or more pounds of P2O5 per acre in that above ground material. And if you assume that 70% of that's going to be contained in the grain that's removed, obviously considerable P export can occur off-site in the grain when you have high pulse crop yields. The soybeans we note here, not too far behind, and at 3 of the 4 sites, the amount of phosphorus in the soybean grain and straw was pretty closer, in some cases even a little bit higher than what we saw in the peas. And indeed, we found the soybeans did have higher phosphorus concentrations in the grain, so we do need to be aware that with soybeans we do have a potential for a greater export of phosphorus perhaps than some of the other pulse crops, and a greater potential for phosphorus down the road because of that higher crop removal. Faba beans were interesting, and I just wanted to bring a little bit of this data in that showed that the faba beans removed about 20 to 35 kilograms of P per hectare, or expressed as P2O5, 45 to 80 kilograms of P2O5 per hectare in the grain alone in Serena Klippenstein's 2016 field trial work. And as you can see at these sites here, folks, again, like nitrogen, a very high proportion of that phosphorus in that above ground faba bean material, a very high proportion of it contained in the grain. Very little left behind in the straw of that faba bean. So really then recognizing again, phosphorus removal in the grain of high yielding pulses can be considerable. And for that reason, we do need to keep an eye on the phosphorus balance, the potential for depletion of soil P fertility over the long term if we're not replacing what we're removing with fertilizer or manure external additions or inputs of phosphorus to balance off that crop removal of P. So a question, what are the effects of legumes on short-term phosphorus nutrition of a following crop, on the immediately following crop? Well, for immediately following crop, we generally observe that phosphorus uptake by a non-legume generally increased following a legume versus a non-legume. And this was observed in some work that was done looking at wheat growing on pulse stubble versus other stubble types, and also for forage crops as well, growing on a forage legume. And some of the reasons why we do see increased uptake of phosphorus when growing on legume stubble compared to other crops can relate to increased soil phosphorus availability due to that mobilization, for example, at depth and bringing phosphorus up to the surface. Also, pulse crops are highly mycorrhizal, and this can translate into an increased colonization of roots by crops that immediately follow, and overall generally improved crop root growth and demand as a result of that legume contributing to that healthier soil and environment. And I think we saw some evidence of this in a study that we did on some of Guy Lafon's plots that he had a few years back at Indian Head, comparing short-term versus long-term no-till, and then different stubble types. And we went out and measured phosphorus supply rate in the field on these sites. And although not sometimes statistically significant, as shown here, we did see a trend towards higher phosphorus supply rates on the pea stubble compared to the wheat stubble, which may be a combination of a number of factors there related to the impact of that pea on perhaps mobilizing, releasing a little bit more available phosphorus compared to the cereal stubble. Moving on past phosphorus then, just a few words on potassium. Some of the data on potassium uptake in pulse grain and straw from Jingzi's study in 2014. Lentil, pea, and soybean at our 4 sites. We've got potassium in the grain on the bottom, potassium in the straw on the top in red. A higher proportion of potassium in the mature plant tends to exist in the straw rather than the grain, in contrast to nitrogen and phosphorus, where most of the phosphorus and nitrogen tends to reside in the grain rather than the straw. So although a lot more of the potassium can be recycled because of return of that straw back to the field, we do see, for example, as shown here, that those soybeans do have more potassium, as much or more potassium contained in that grain and straw compared to the other pulse crops. So indeed, soybeans are relatively higher potassium users, and also a potential there for greater potassium depletion as a result of grain removal when soybeans are growing. And finally, sulfur. Just to share some of this data as well, sulfur uptake in the above ground plant material, the grain and the straw. Sulfur is a little bit more evenly distributed between the grain and the straw, oftentimes about 50-50, half of it in the grain, half of it in the straw. And we can see here that not a lot of difference amongst those different pulse crops. And again, the soybeans showing up sometimes as a little bit higher sulfur requirements, sulfur user, and grain removal of sulfur sulfur as well compared to the other two pulse crops. This pea sulfur uptake, very high sulfur uptake at the Saskatoon site, was kind of unusual, and we questioned why this might be the case. And then coming back, recognizing that this is a site that has a lot of sulfate in it because of the salinity, a lot of sulfate at the surface early on in the season when the water table is high. And what we think happened here in the case of the peas at the Saskatoon the site is that they took up that sulfur earlier on, and there was more available in there earlier on, and so we see that manifested in a quite a bit higher sulfur uptake at by the peas at the Saskatoon site compared to the other two crops. So I guess, to conclude, folks, certainly when it comes to nitrogen, we observe and recognize that pulse crops can largely meet their nitrogen requirements and indeed balance off that crop nitrogen removal through fixation of atmospheric nitrogen. And that's one of the real fertility value benefits of pulse crops. But recognizing as well, when we think of the other macronutrients, phosphorus, potassium, sulfur, certainly considerations for depletion of phosphorus, potassium, sulfur are important, especially when you're getting high yielding pulses. They're growing in rotations for several years. So I think what that means or boils down to is that really when we're thinking about good pulse crop fertility management, we really need to be thinking both short-term and long-term. How does the pulse crop influence the nutrient requirements for the immediately following crop, that is to say short-term, but also what are the implications of having that pulse crop in rotation there over the longer term? And I guess the tools that we have to do that for short-term requirements are your soil test to look at what's required for that crop that you're going to grow this next season. But over the longer term, certainly considering the nutrient removal of those different pulse crops, especially what's removed out of the system in grain and relation to what's being put back into the system, for example, through fertilizer, so that you can keep tabs on what's happening to your nutrient bank account. Are you depleting soil phosphorus fertility? Are you maintaining it? Are you building it? All a matter of balance over the long term. So with that, I would like to thank Jing Zi and Serena Klippenstein, the two students who work on these pulse nutrient requirement contribution projects, Dr. Tom Warkentin and Dr. Bert Vandenberg, who I work with as well on the pulse crop fertility nutrient requirement projects and the Crop Development Center Pulse Crew, and our funders, Saskatchewan Pulse Growers, Agriculture Development Fund, and the Western Grains Research Foundation. So with that, I will conclude and turn it back over to you, Andrea and Glenda.
Thank you so much, Jeff. We really appreciate your time and your efforts to help us out with the webinar today. For those who are listening, please, we still have time. We have a couple of questions in, and if you have come up with any questions, you still have time to enter them so we can ask Jeff your questions. Just please type them into the question box on the GoToWebinar. So Jeff, a couple of questions that we had was, you had mentioned a bit of information around safe placed rates for phosphorus. Do you have any recommendations around for fertilizer placement where it comes to perhaps phosphorus and potassium combined, as well as sulfur, and the best way to make those applications?
Yeah, and I think we did some work a few years back looking at phosphorus alone, that is monoammonium phosphate, 1152 alone, and then 1152 plus a potassium source, potash. And potash, KCl 0060, has a high salt index. And so what we found, in fact, when we had our 15 of our phosphorus, P2O5, and then we put on another 10 of potash, KCl, we significantly reduced the emergence and germination. So yeah, when you start to add other things in there in addition, and ammonium sulfate would be another example of a fertilizer that has a relatively high injury potential, not only from a salt effect, but also from an ammonia toxicity effect. Yeah, when you start to lump other things on top of that phosphorus in there, indeed, you can end up with some significant reductions in germination and emergence. So I think a person needs to be aware of that, and probably, you know, if you're looking at, for example, sulfur, having it in a separate band is the place to put that sulfur rather than having it in a seed row. I really don't think a person wants to run the risk of having a poor density, a thin stand because of injury of thinning from too much fertilizer placed with the seed row. I will say that we are starting a new study to take a look at tolerance of the different pulse crops to different starter nutrients, including nitrogen, phosphorus, and sulfur. These include both combination fertilizers and blends of more mainstream granular fertilizers. So we'll have some better answers, I think, to that in a couple of years down the road.
Excellent. Thank you, Jeff. And another question. If growers have crop remaining in the field, such as lentils this spring, how would you recommend that they manage that crop perhaps, and how would that impact the fertility of the field based on what they choose to do to manage that crop?
Yeah, and I guess if the lentils are going to remain unharvested, you know, and they might end up being seeded directly into with another crop, or they may end up being tilled or worked into the soil. I think one of the things to keep in mind, and I guess this is maybe a little bit of a bright side to it, as I explained, the bulk of the nitrogen is contained in the grain, maybe 3/4, might even be higher than that in the case of a faba bean. But in a lentil, maybe 3/4 of that nitrogen is contained in the grain. It wasn't harvested, so it's there in that grain, and that nitrogen in that grain, when that grain starts to decompose, available nitrogen will be released. So it's an additional contribution of nitrogen in that unharvested crop above which you would see just from in just harvested pulse stubble alone. The rate at which that nitrogen is going to be released, it's going to be released gradually over the season. I remember one study we did where we looked at the release of nitrogen from pea grain, and actually it took place gradually, but it started out right away in the spring. We started to see some release of nitrogen, available nitrogen, because the grain has quite a high nitrogen content, a narrow carbon to nitrogen ratio. So it won't immobilize available nitrogen initially like straw will. So yeah, there's an additional contribution that's going to come from the unharvested grain that remains in that field. How much can you expect from that? It's going to be difficult to say. It will depend on whether it decomposes more slowly on the surface or if it's worked in. That whole decomposition is probably going to take place faster. And the other factor is going to be the environment. If it's moist and warm, that release of available nitrogen from that unharvested grain will take place more rapidly.
Thanks, Jeff. A few more questions are coming in now. What would you say is a healthy number of nodules on a pea plant?
Boy, I don't know whether I have the answer to that or not. I mean, oftentimes we do counts, you know, nodule number. You can cut them open and look inside and you know, are they that nice bright pink color? But, you know, just because there's nodules present doesn't necessarily mean that they are actively fixing nitrogen to a high degree. We can have nodules that are formed, but they're relatively inactive. So nodule number, I think, is perhaps not the best indicator of fixation. But I mean, you should see some nodules. You need to have those nodules on that root in order for fixation to take place. And as I showed you in my presentation, those roots there where a lot of nitrogen fertilizer was added, you look at those roots, you can't find a nodule on them. There's little or no nitrogen fixation taking place there.
Okay, thanks. And when making fertilizer recommendations for crops grown after pulses, how many pounds of nitrogen credit would you use on average?
That's a good question, and I guess one of the interesting things that, you know, yield of the previous crop, I think works pretty decent, but I think what's interesting in the work that Jingzi did in her thesis was that probably the straw yield if you could measure it, was more closely related to the differences that she saw among crops in nitrogen availability than the grain yield. But straw yield is something that's not very easily practically measured. Grain yield, of course, is something that is. And so I think it's a pretty good indicator. And you know, values out there in the literature I've seen range anywhere from maybe a half a pound of nitrogen per bushel of yield to upwards of 1 pound of nitrogen per bushel of yield additional credit. And I think those differences that you see out there in the literature probably reflect just differences in maybe proportioning, depending on the crop, depending on the conditions, differences in allocation or proportioning of the nitrogen between the straw and the roots, maybe even in the decomposition rate. But you know, I would probably say, probably maybe, you know, I probably tend to maybe go, you know, you could split the difference and say maybe 3/4 of a pound of nitrogen for each bushel per acre per yield might be kind of an intermediary estimate for most pulse crops. But yeah, I've seen studies a half a pound to upwards of 1 pound based on some work that was done out of Alberta.
Thank you. The next question is, what is the maximum safe rate of seed-placed fertilizer, particularly in sandy soils or low organic matter soils with pea crops? And are there differences between soil types?
Yeah, I mean, I think in general that when it comes to the maximum allowable seed placed rates that you see, you know, out there, for example, in the provincial guidelines here in Saskatchewan, assumes good moisture, medium type textured soils. That's where the work, for example, loamy textured soil that we did the work with the soybeans on, about 3% organic matter. Generally, higher clay content, higher organic matter content tends to reduce the toxicity from the seed-placed fertilizer. Also higher moisture content. So generally we expect to see injury first and worst on sandy textured soils. Dry soils, also high pH can aggravate injury because the high pH tends to favor free ammonia. So if you're using ammonium sulfate in the seed row with a pea and the pH of the soil is high and it's sandy and it's dry, only a very small amount of ammonium sulfate may be all that's needed in order to produce significant injury. So, you know, I almost think that under those conditions where it's dry and it's sandy and high pH, you know, to try and keep that phosphorus, if possible, maybe out of that seed row or a very low amount, you know, and put it in a separate band.
Thanks, Jeff. So I'm going to ask the next question, and my apologies if I'm not pronouncing it correctly, but it was a question around your opinion on a phosphorus source for pulses, in particularly Mez-Z. Is that something familiar to you, Jeff?
Oh, must be MES?
MES?
Zinc.
Yeah, okay.
Or
yeah,
zinc.
Yeah. So yeah, so that's one of these, I guess, what I refer to as a combination fertilizer, sometimes referred to as a specialty fertilizer, where you've got nitrogen, you've got phosphorus, you've got sulfur, you've got some zinc there. So yeah, that's an example of a fertilizer that provides not just phosphorus but also some nitrogen as a starter, some sulfur, and even some zinc there. The interesting thing that we've observed so far, and we haven't done a lot of work with this, but with pulse crops, but we have done a little bit more with canola. But with some of the combination products like the MES, and there's other ones out there as well, there does seem to be slightly less injury potential from those combination products compared to where you're just blending together, for example, ammonium sulfate and monoammonium phosphate, and that's what we found in some of our recent work, and we're going to be exploring this more in detail in a new project that we have that we're going to be doing over the next 2 years. But yeah, some of those combination products that are out there do seem to be not quite as injurious, and therefore can use slightly higher rates with the seed compared to blends of some of the conventional products.
Do you have a comment on the zinc that's in there, or zinc as well?
Yeah, and although I realized I couldn't cover both micronutrients and macronutrients in this one presentation in an hour, and we have been doing some work with micronutrients in pulse crops, and that's, I think, another story for another day. But I will comment on the zinc aspect, and we did some work with lentils, and more recently with peas. And generally what we see is small and variable responses to zinc fertilization in the pulse crops we've worked with so far, which have been pea and lentil, and also responses that are not really well correlated with our soil assessments of availability. There's some relationships there, but sometimes we've seen scenarios where a soil will come back as predicted very zinc deficient, and yet we didn't see any response to added zinc fertilizer in field trials. And I guess that's kind of the nature of micronutrients in general, is that they can be kind of difficult to predict the likelihood of seeing a response to, and under some conditions the deficiency symptoms can arise, and then environmental conditions change and they disappear. But I would say, in general, in the work that we've done so far, that we maybe only encountered a couple of soils in a range of soils that we collected from across the prairies where we saw the pulse crops, and I'm thinking here particularly of pea, respond in a big manner to the addition of zinc. One of the problems with zinc that's soil applied is that it does tend to, in calcareous soils, tend to become quite readily fixed. And so one of the things that we did observe is that it seemed like, at least in some of the work that we did with lentils, that the foliar applications were somewhat more effective than the soil applied. I think because the soil applied zinc in the calcareous soils with lots of carbonates, we were getting a fair bit of that soil applied zinc that we were broadcast and incorporating fixed as carbonates.
Thanks, Jeff. Another question. Assuming you can only seed place 40 pounds per acre of P205, if you're growing faba beans in rotation, what would be some suggestions for keeping the phosphorus balance in the soil?
Okay, no, that's a great question. I think probably you'd look at upping. One option would be upping the rate that you're applying in the crops that you're growing in rotation with faba beans. So if you've got a cereal in there like barley or wheat, those crops are tolerant to 40 or 50 pounds of P205 per acre in the seed row. So you can be upping your rates with those crops. Other alternative to that would be putting phosphorus in the seed row with your faba bean, but then also putting some phosphorus in a separate band, a side band, a mid-row band is another effective strategy as well, I think. Looking at what we saw from broadcasting, just spreading it on the surface in very phosphorus deficient soils, just broadcasting without incorporating, we found that to be not a very efficient method of placement of phosphorus, because I think what's happening there is when you're broadcasting it on the surface, for example, especially of a calcareous soil, that phosphorus that you're broadcasting is just hung up there on the surface, and it ends up being too far away from the roots to get to it, and so is not utilized very effectively by that crop or even the crop that would follow in rotation.
Thanks, Jeff. We have one final question. Just if you could comment on nitrogen credit to crops following alfalfa.
Okay, yep, and that's now we're into forage crops, which is great because we've also done some work on the forage crops, and as I said, I gave some nitrogen replacement values on one of those slides of I think 300 kilograms of nitrogen per hectare. That's a calculated value, and that's a nitrogen replacement value. How much nitrogen did you need to add to make those curves that I showed in the previous diagram converge? So that's a lot more than what the actual direct nitrogen credit is from alfalfa. So I would say probably, you know, what you might expect from a good alfalfa crop as a direct nitrogen credit, let's say from a 5 ton per acre alfalfa crop, you know, might be maybe more in the order of maybe around 150 pounds of nitrogen per acre to the following crop. And it's going to depend as well on how that alfalfa is terminated. Some work that was done by Dr. Molly in the northeastern part of Saskatchewan showed that when that alfalfa was terminated with a herbicide, it tended to slow the release down, such that less was released in the first year, but conversely, more tended to come out in the second year. So it was in the total amount that came out was about the same, but that when you delayed when you terminated with herbicide, it tended to slow down or spread it out over a longer time period. But yeah, some of the numbers that I've seen out there in terms of direct contribution, you know, might be closer to that, you know, 150 versus some of the calculated nitrogen fertilizer replacement values that I showed there that were, you know, upwards of, I think at the Melfort site was around 300. So I think nitrogen credit from alfalfa in general can be anticipated to be, you know, somewhat higher than the nitrogen credit you would see from an annual legume crop, a pulse crop.
Thank you very much, Jeff, for speaking to us today. Also, thanks, Andrea, for organizing the session today, and a very big thank you to all of the participants for joining us today. Please mark your calendars for our next webinar, which will take place at noon on May 15th, covering soybean agronomy. As a reminder, you can sign up for this webinar from the SPG website under News and Events. And also a final reminder for CCAs on the call, and that an email will be sent out to you, and you will need to reply with your CCA number in order to get your credits. Thank you very much, everyone, and have a great day.